So to start the session, just a little bit of an overview of the talks that you're going to hear about. This is a figure from a very nice review by Schlocke just earlier this year. And as you can see on the left, active immunization, as you all know, induces immune responses upon exposure to an antigen or a pathogen. And then there are different kinds of passive immunity that can be achieved in individuals. In the middle is the more classical kind of passive immunity, and that's where protein is delivered, recombinant antibodies typically. And then on the far right is sort of this new phase of passive immunity that's going on right now. And this can be achieved through the transfer of nucleic acids or through the transfer of T cells. So today you're going to hear a talk by Dr. Protzer on passive immunity provided by T cells for HBV. And then today I'm going to be describing DNA delivery of antibodies. Dr. Ballas will describe delivery of antibodies with viruses. And then Dr. Weissman will talk about mRNA delivery of antibodies and other proteins too. So as I just went over, there are a number of ways to provide passive antibody immunoprophylaxis. And as you all know, recombinant proteins are very effective, but they're expensive and they require frequent administration. And so there's a real advantage if there can be the development of nucleic acid-based approaches for providing antibodies to protect from disease or to act on endogenous targets also. And these can be in a number of forms, like as we just discussed, viruses, and mRNA or DNA, and all of these methods have pros and cons to them. Yeah, and what I'll discuss today is delivery of DNA using technology because DNA just injected on its own doesn't provide high levels of protein expression. There isn't good entry of DNA into cells, and so delivery technology, electroporation, is what I'm going to focus on today. And this isn't a new finding. There were two papers published way back in 2004 on delivery of antibodies using electroporation-mediated entry into cells, and this was with plasmids encoding antibodies. But this field didn't really take off. This was more a proof of concept at that point, just to show that, that there was a powerful technology with electroporation being able to get plasmid DNA into cells. And the field, however, kind of moved more towards other applications of plasmids at the time. But this is the basic principle, that electroporation, when it is used to deliver plasmids that code for an antibody, the plasmids enter muscle cells, and the muscle synthesizes the antibody. It's secreted, it enters the bloodstream, and then the hope is that the antibody in the bloodstream, and as it distributes throughout the body, that it can protect from infection. And so what the idea here in particular is that with conventional recombinant antibodies, they have to be frequently readministered. And so there's a PK where you get these spikes, and after about a month or so, you need to readminister. Whereas theoretically, the hope is that with nucleic acid-based delivery approaches, especially DNA-based approaches, you can get more of a long-term expression, so readministration will be less frequent. So our company became interested in this approach primarily because of a program that DARPA, which is the Defense Advanced Research Projects Agency, it's an agency of the Department of Defense. They came up with this program because they're interested in high-risk, high-reward programs that can help, in particular, troops, and also it's very nice of their civilian applications also. So in a pandemic, troops and medics need rapid protection. And as you can see from this map here, this is an example of what happened in 2009 with the pandemic influenza infection, where very rapidly this H1N1 spread around the world. And so if there are troops in areas of instability or there are medics needed, if there isn't a vaccine that can protect from disease already, then there has to be another approach to protect the safety of the military. So this is why DARPA became interested. And they selected a nucleic acid platform for antibody delivery of immunoprophylaxis for a number of reasons. One of the main ones is that there's a single manufacturing process that enables a rapid response. So unlike recombinant antibodies, where there's a different purification approach for each antibody, if you're making plasmid DNA that codes for antibodies, the manufacturing process is always the same. So you can have a manufacturing approach that's ready to go. DNA has an excellent stability, profile. There's the potential for infrequent dosing, which would be particularly important, and a potential for reduced costs compared to development of vaccine or compared to recombinant antibodies. And so our company was awarded about $11 million to investigate DNA-based antibody delivery. Just to tell you a little bit about our electroporation technology, it's called the Trigrid Delivery System, and we've been around for a while. We have more than 30 DNA vaccine trials that are either ongoing or have been completed, and these are in a number of continents around the world. We're in phase 1 and phase 2. The procedure has been administered to over 800 subjects with more than 4,000 administrations at this point. And I'm not going to go into it more, but if anyone wants more information on safety, which has been excellent, and tolerability, you're welcome to talk to me after the talk. Basically, what we've shown is that electroporation does work in humans to increase the delivery of DNA into cells. And we've been able to show that there's improved potency versus conventional injection in a number of trials that were randomized and placebo-controlled. We've shown that DNA vaccines can elicit cellular and humoral immune responses, and we've also shown that there's preliminary indication of functional disease or activity modulation. And so it's in the process primarily focused on DNA vaccines. It's a very good application of the technology to use electroporation with DNA vaccines. However, we've also been interested in the delivery of proteins, and this is some data that we generated a while ago now. I can't believe it was 2006. But what we wanted to show is that a single DNA administration encoding a therapeutic protein, in this case interferon beta, could be as effective as repeated injection of a therapeutic protein. So in this case, just one figure from this paper where interferon beta was delivered to a relapsing remitting EAE model, which is a model of multiple sclerosis. And what you can see in the upper graphs here is that in this model, if the mice aren't treated, they get progressive clinical scores over time. And so the green group received a plasmid that didn't code for a protein with electroporation, and the red group was just the untreated group. But if you look at the groups down below, the blue group received a plasmid that encoded for interferon beta. It was delivered at a single time point, so at the very beginning of the clinical signs, so at day 0 on this the x-axis. And then the black graph there shows what happened when the mice were treated every other day with recombinant interferon beta. And what you can clearly see is that the plasmid-based delivery of interferon beta had the same effect as 30 administrations of recombinant interferon beta. So we had this proof of concept from years ago that delivery of plasmid DNA encoding a protein could have sustained disease-modifying effects. And so moving into this DARPA program, we had some confidence that we would be able to express antibodies long-term with electroporation-mediated delivery. So first experiments we did were to show that electroporation is essential in order to see antibodies in the bloodstream. This is two sets of experiments where we encoded a Rhesus monoclonal antibody specific for the F protein of RSV. And on the left panel, you'll see the results with mice. On the right panel, the results with rats. And what you can clearly see by the blue bars there is that antibody could be detected in the microgram per mil range when electroporation was applied after DNA delivery, whereas no antibody was detected in any of the animals when electroporation was not applied. And in this experiment, the heavy and light chains were encoded on separate plasmids. It was a CMV promoter enhancer from a commercially available vector. And we delivered 10 micrograms in the mouse or 20 micrograms in the rat to a single muscle. Oopsie. So then next we wanted to look at the duration of expression. And in this experiment, this set of experiments, you can see the duration in mice and in rats. In mice, this was a monoclonal antibody specific for anthrax protective antigen, and for rats, it was a rat RSV specific antibody. And what we saw was peak levels typically around 2 weeks or so, and in these experiments with about 2 to 5 micrograms per mil at peak that then kind of settled down to the 1 microgram per mil range, and there was sustained serum presence for more than 6 months. So next we wanted to know if you could scale this up to a non-human primate to see if this finding of sustained expression could be recapitulated in not only a larger species but also in a primate species. So in this experiment, we delivered plasmids coding for a Rhesus RSV-specific antibody. And what we did was we delivered 3 mgs of DNA to each quad muscle. So it was a bilateral delivery of both the H and the L chain plasmids. And the red bars indicate monkeys that were about 11 to 15 kilograms. And the purples, there were 2 younger animals, and these are about 3 kilograms. And what we were very happy to see is that we were able to see sustained expression of the antibody in sera from all the monkeys except for one for greater than 3 months post injection. So a single injection of DNA with electroporation at day 0. I can't get the pointer to work well, but you get the point. And then a sustained expression through past 3 months. And I'm going to talk more about the one monkey that showed a decrease in expression before day 60 a little later in the talk. So we were surprised to see sustained expression for so long, and we continued the experiment, and again, much to our surprise, we saw in the larger monkeys expression go away at about 7 months, and those are in the reds there. And in the 2 younger monkeys, we could still detect antibody in serum more than 1.5 years after administration of the plasmid DNA. And we wanted to be confident that the plasmid, the antibodies we were detecting in the younger monkeys really was the RSV-specific antibody that we had administered to the monkeys. And so we made anti-idiotype antibodies and confirmed that there still was antibody detectable in these individuals at very low levels at this point, of course, but still very interesting to see that more than 1.5 years after administration of the DNA, there was still antibody being made. So we wanted to know, can you re-administer antibody and see a bump up to peak expression levels again? And so we delivered plasmid encoding a murine antibody specific for the anthrax PA on day 0, and then re-administered the plasmid at day 200, and we did indeed see a bump up in expression, showing that you could get this re-administration effect. And then we also wanted to know, can you express more than one antibody from the same animal? And so in this experiment in mice, we encoded the H and L chains of an RSV-specific and an anthrax PA-specific antibody on 4 separate plasmids. And we either delivered the plasmids for the RSV antibody or the PA antibody to separate muscles, or we mixed them all together and then spread them between 2 muscles. And what we saw was that with the RSV-specific antibody, we saw about the same levels of antibody in the serum, regardless of whether the plasmids were mixed or separate. And so this was a surprise to us. We hadn't really anticipated that we wouldn't see much of a difference. With the PA-specific antibody, there was more of a difference between them, but you could still detect antibody even when the plasmids were mixed. So this indicated that it is possible to produce multiple antibodies either by delivering to separate muscles, and with appropriate antibody engineering, it might be possible to get expression of multiple antibodies from the same muscle. So we looked at a number of approaches for increasing the serum levels. What I've been showing you, typically our best expression is up to maybe 5 micrograms per mil, typically maybe 1 to 2 to 3 micrograms per mil. And we did a number of evaluations of plasmid backbone and regulatory sequence and modifying antibody sequences and looking at formulation. And what we found really was that we're very good at decreasing expression by modifying the plasmid backbone and regulatory sequences. It was hard to get anything that actually increased expression, so I'll just admit that publicly. It's hard to admit, but we did a ton of work and really didn't find anything better than we were already using that was more than about 2-fold better. Modification to antibody sequences definitely could increase antibody levels, sometimes 5 or 6-fold, but they were antibody dependent, so there wasn't any one thing that we found that always worked for every antibody. Tried a number of formulations, and the only thing we found is pretty consistent with what others in the field are seeing also, is that if you co-deliver with an enzyme called hyaluronidase that degrades hyaluronic acid in the extracellular matrix, then you can get an enhancement of antibody expression. And an example of that is shown in this slide here, where this was an experiment in mice where we delivered antibody either without hyaluronidase, so these were plasmids encoding antibody, or with hyaluronidase, using either the bovine version or the commercially available Hylenex that's already marketed. And one concern we have with co-delivering hyaluronidase with antibody DNA when delivered in the context of electroporation is that there are already is data showing that Hylenex or hyaluronidase is immunogenic in humans, with about 2 to 18% in various trials of incidents of antibodies that have already been described. And so there is this potential safety concern that delivery of DNA with electroporation could enhance these autoimmune or ADA responses to hyaluronidase. So it's just something that will have to be monitored. And what's very exciting is that Inovio recently initiated a trial where they're evaluating electroporation-mediated delivery of plasma DNA encoding a Zika-specific antibody with hyaluronidase. And that will be a really, really important marker for our field to see whether antibody can be detected in individuals, and looking at the safety profile also. So some of the vector development we did, just to show you, one thing we really want is to have a plasmid that codes for both antibody chains, and we've had a hard time finding one that works as well as coding the antibody chains on separate plasmids. And this is an example here of where we tried a commercially available vector from Oxford Genetics and compared it to a dual gene promoter vector that we had developed internally that has CMV and CAG promoters, and then compared it with encoding the chain separately on GWiDs. And here you see about the same level of expression. With dual gene plasmids, it's been hard to see higher expression, which you might expect there might be on a dose, on a mass basis. If you deliver the same mass of DNA, whether there are 2 plasmids or 1, you would hope that you'd see twice as much expression, at least with the dual promoter or the 2 gene versus the 2 gene plasmids. But typically, it's been hard to get good expression. So this is about as good as we've gotten so far. There's huge manufacturing advantages to have them both encoded on the same chain, and so that's our goal, is to continue this kind of development. So a really important aspect of this work is whether the antibodies are functional, and our collaborators at the Aaron Diamond AIDS Research Center in David Ho's lab did some experiments, and they were published in 2017 by Andrews et al. Really nice, elegant experiments. Just one example shown here, where they encoded 3 influenza-specific antibodies on plasmids, delivered them with our TDSIM electroporation device, and they showed that when the mice were challenged with influenza, that they could see protection compared to 100% death in the case of the animals that received control antibodies. And they've used this for other challenge studies also, some very elegant work. But bottom line is that the work done by David Ho's lab and also out of David Weiner's lab, his group has published a number of studies showing that DNA encoded antibodies are indeed functional and can protect from disease in a number of disease models. So wanted to describe one of the things we've seen is that it's very easy to get host responses against stenotogenic antibodies. So on the left panel here, we have an expression of a rat RSV-specific antibody where we get long-term expression. On the right, we've graphed individual animals and we've expressed a Rhesus RSV-specific antibody. And what you can see here is that it kind of looks like we forgot to graph the bars at day 10 for the individual animals, but we didn't. At day 10, there was absolutely nothing detected in these animals. And what happened is that we have an assay to detect host anti-monoclonal antibody responses, or what we call AMA responses. So in the case of the rat, there's no antibodies detected because you get long-term expression. The rat is a self-protein. On the right panel, you can see that all of the animals developed AMA responses against the antibody. And this isn't a surprise. This is what you would expect for a xenogenic antibody. But when we saw that kind of data, it made us think when we did this monkey study about this animal right there, where the expression went down pretty quickly and long before the other animals, and we decided to investigate this further. So the first thing we did was we re-administered the DNA at day 98, and we did not see a bump up in expression, and so this was concerning. And when we did our assay to look for anti-monoclonal antibody responses, we did indeed detect anti-monoclonal antibody responses at quite high levels after the second DNA injection. So we've also seen this in other NHP studies. And what is very interesting to us is that recently there were clinical trial results published for an AAV HIV monoclonal antibody trial. It's a PG9 antibody, where no antibody was detected by ELISA in the serum of humans after delivery of the AAV PG9 antibody. vector, and 21 out of 21 subjects had anti-AAV antibody responses, but what was really interesting is that 10 out of 21 also had anti-PG9 antibodies. This was published in 2019, just recently in Lancet HIV. And so it's clear that this observation we've seen in the monkeys was also seen in an antibody trial in humans. And so understanding the factors that contribute to host anti-monoclonal antibody responses is going to be really important to the future of this field. So we were really hopeful when we started this project that we would get really high levels of expression. Clearly we've got work that needs to be done, but DARPA was convinced enough by the initial work that they did another program called the Pandemic Prevention Platform, or the PRE3 program. And in this program, what they want to do is to develop a functionally integrated platform to deliver protection against a pandemic, a newly developing pandemic, in less than 60 days, which is really pretty crazy if you think about it. They want to go from antibody discovery through to, having doses. They're preferring 20,000 doses ready to administer to humans in 60 days. And when those of you that are in the development process know that normally that would take probably 10 or 15 years to do. And so DARPA likes to do these science fiction sort of projects, and we were fortunate enough to go in to a program with a Canadian company called Accelera. They're an antibody discovery company, and they are able to develop very potent antibodies. They discover them from humans primarily, but also can do it from a number of animal models. And so Accelera is doing the antibody discovery portion of the P3 program that we're a part of, and then we are doing the encoding on plasmids and delivery. And what we're hoping is that with really potent antibodies, even though we don't get the higher levels of antibodies, say, that viral delivery can achieve, that with a potent antibody, maybe there's enough in the serum that would be okay. And this is something that DARPA is definitely very interested in. And then primarily, what's been really good about the DARPA program is that DARPA's investment in this nucleic acid monoclonal antibody delivery technology is that it has spurred pharma interest. And so, for example, we recently publicly announced a collaboration with AstraZeneca, where they are interested in using our delivery technology to deliver antibodies encoded on DNA. So to sum up then, what I've shown you is that we can see expression of monoclonal antibodies detected from muscle for greater than 3 months, and it's usually much longer than 3 months in mice, rats, and in non-human primates. And that these antibodies can protect from pathogen challenge, so they are functional, and therefore this really seems like it could be a platform that could be used to protect against infection. So in order to get protection in humans, we're going to need potent antibodies, and in addition, we're still looking for ways to increase serum antibody levels. And it's clear from the recent trial with the AAV vector and also our monkey studies that there have to be strategies to reduce the incidence of host anti-monoclonal antibody responses. And in addition, we need to have an understanding of potential safety issues. For example, if you express an antibody that has off-target binding. Now, there are real advantages to this platform. They include single rapid manufacturing process and a potential cost savings, and could be more of a rapid deployment, not only to troops around the world, but this technology could also be used in developing countries where the use of recombinant antibodies is really inhibited by the cost. So I guess the question is, will nucleic acid encoded antibodies be a disruptive technology? Could they replace the use of recombinant antibodies in some cases? And I think we really don't know at this point. There are a number of hurdles we're going to have to cross. Primarily, I think, is the level of expression, and then this finding of potential immunogenicity of antibodies. But we are hopeful. I'd like to say a big thank you to the ICOR team, who has worked really hard on these projects. Also, thank you to our collaborators at Aaron Diamond and David Hoslab and O'Neill Patty's sitting over there. And we really appreciate the funding that we've had from DARPA over the years. Thank you.
So this paper is now open for questions.
Hi, Claire. That was really nice. I guess I'm wondering, do you have any idea what the mechanism of the longevity of expression is in your mice that are expressing for like a year? I mean, have you looked for integration?
We have. We've had to look at integration. When we first started our first DNA vaccine clinical trial back in 2007, the FDA was really worried about potential integration of plasmid DNA into the host chromosomes, and so we developed integration assays and have actually done numerous GLP studies, and we really find only very, very low levels of, we don't even find levels of integration. We find maybe a hint that there could be some very, very low levels, but it doesn't appear that it's integration. It's persistence of the vector. The muscle cells aren't turning over in general, and so it's probably an episomal event, but it's surprising, but
And I guess my other question is, did you guys ever try passive transfer of the same Rhesus antibody in monkeys to see if the protein itself was somehow slightly immunogenic?
Yeah, we haven't done that, but our collaborators have, so you could talk to Neil Padty about that. But in general, the results from us and what other people have done also is that the problem is when you do, say, IV transfer of an antibody, it actually isn't, I don't know what the right word is, it produces some level of tolerance. It like floods the system, and so I don't think the antibody is as immunogenic, because certainly proteins that aren't very immunogenic can be administered IV, and then when we produce them in muscle, they are more immunogenic. And part of that probably is because we're introducing DNA plus electroporation, which causes tissue damage, so we have an adjuvant-like effect, so there's probably more of a propensity, but I don't think just by comparing the IV versus the muscle, the endogenous expression in muscle, you can't really predict immunogenicity based on that sort of test. Thanks.
Yep.
What's on? Sorry about that. So Claire, I was wondering what's the ideal titer you'd like to reach? You said 5 microgram per mil, and whether it could be reached by dosing at multiple muscle sites at the same time.
Yeah, good question. I actually didn't go through that. I had one slide where there was a lot of writing, and I just felt it was too early in the morning to go through all of it. But if you deliver to more than one site, it's an additive effect, and so the more muscle sites you deliver to, the better. The problem is that with electroporation, there is, don't tell my CEO, but there is some transitory pain associated with it. It's very brief. It's less than a millisecond, or less than a second. And so doing multiple sites probably isn't ideal in the case of a prophylactic application. If it was for a therapeutic application, I think that would be acceptable for sure. As far as target antibody levels, it's really hard to say right now, and that's really going to have to be done through studies, especially I think challenge studies in humans, because we don't know if there's a difference in the amount of antibody needed. If you have something like an application IV administration of a recombinant antibody versus this endogenous production, we don't really know whether same serum levels will be needed. However, most people probably think at least 10 micrograms per mil is maybe more, a good goal. And what we're hoping is that with potent antibodies identified by our collaborators, that a lot lower will be acceptable. So, jury's out.
Just a follow-up. So if you achieve higher titers, do you expect more AMA?
That's a good question also. Yes, you could expect more, but on the other hand, what we are thinking is that there probably are some intrinsic properties of some of the antibodies. Because I didn't have time to go into this, but we have actually seen this in some rodent models, and we're trying to understand what features of antibodies could possibly promote these AMA responses more. So I think we need a better understanding of that too.
Just a quick question. Has the TDS been tried like in dogs and cats? But you mentioned bovine, I think, so
yeah,
could comment on other animal models? Yeah.
Yes, it's been adapted to 15 animal species, birds, chickens, llamas, cattle, just about any animal model, ducks, yeah.
Okay, I'll just ask the last question. What's the role of the HLNX in the delivery? The HLNX in the delivery.
Oh, hyaluronidase?
Yeah.
Oh, I'm so sorry. Yeah, I didn't really explain that. Hyaluronidase, it degrades the extracellular matrix. There's hyaluronic acid that makes up the part of the extracellular matrix, and so it's used in humans. The Hylenex product is used in humans to get better dispersal of drugs. And so we think it's probably the same thing. That might be better contact of the DNA with muscle cells upon injection. Could also be helping antibody to get from the muscle cell into the bloodstream, so better dispersal through the tissue.
Yeah. So, yeah. It simply won't stop. There. Okay, thank you very much, and thank you for being on time. So, perfect. Next speaker